Welcome to the Cromatin practical for BS2002. In this practical, we're going to test the idea that DNA is packaged into cells and is wrapped around nucleosomes, which are made of histone proteins. To test this, we're going to use an endonuclease, which will be able to cut the DNA where it is not protected by the nucleosomes. We're also going to have two control experiments. In tube A, which we are not going to use the endonuclease, and in tube C, where we're first going to extract away the proteins from DNA with phenylchloroform, and then we will hit it with the nuclease, which therefore should be able to cut the DNA at any point. The endonuclease that we're going to use is from bacteria. The enzyme cuts at any point along the DNA, unlike an exonuclease, which only cuts from the two ends. You should note that it requires both magnesium and calcium, and if you see in the protocol, at times we use EDTA, that is a chelator of magnesium and calcium. So you should think why we use EDTA. The first step of the experiment involves solubilizing the membrane. Twin 20 is a mild detergent that will dissolve both the cellular membrane and the nuclear membrane so that the nuclease can then enter to the nucleus. The phenol extraction is a crude way of removing the DNA or separating it from the rest, and so that the DNA will end up in the aqueous phase. Phenol does not mix with water. We can force this mixing by vortexing, but when we centrifuge the tube, then the two phases will reseparate and the DNA will be in the aqueous phase and everything else will be in the phenol phase. We then carefully transfer the aqueous phase to a new tube, to which you will add chloroform, which also doesn't mix with water, and repeat the extraction. The chloroform extraction is really to remove whatever excess phenol may have been left. The samples will then be loaded on a gel, on an agarose gel, and through electrophoresis, DNA, which is negatively charged, will travel towards the positive charge. The gel contains a dye that allows us to visualize the DNA under UV light. The results we're expecting is to see high molecular weight DNA in lane A, a 200 base pair size DNA in lane B, and completely digested DNA in lane C. The molecular weight that you see are used to try to estimate the size of a bands that you see. In this practical, you're using phenol. You must be very careful when you use phenol because it's corrosive, it will burn your skin and damage your eyes if you come into contact with it. So use gloves and be very careful also to always cap any tube that contains phenol before discarding them. Do not put them anywhere uncapped. Also be careful with chloroform, which is toxic.